Hello to our friends joining us via recording. We are reviewing for the lab midterm exam. As a reminder, that exam is open right now on Monday, October 5th, and it closes tomorrow night at 11:59 p.m. Tuesday. So make sure to plan some time for yourself to sit down and take that exam. Big part of the lab midterm exam is being able to identify tissues in pictures and being able to do things like naming what their function is or what their location is. For some types of tissues, naming the types of cells that we find inside of them. So this is a review activity to help you recognize that one of the locations that we find a lot of the different types of tissues that we're learning this semester is different parts of the skin. So the image we're looking at here is a cross section of the skin from the outside up here at the top down to the very inside, the deepest layer of the skin. So the outermost layer of the skin is called the epidermis. Epi means above. The epidermis is above the dermis. Remember from your studying that the dermis has two parts, the part that's up here at the very top called the papillary layer and deeper layer below it called the reticular layer. Both of those are part of that area called the dermis. Underneath the dermis, we get into an area called the subcutaneous layer, or the other word that you'll see this area called, let me add it for us, is the hypodermis. Hypo means below. So the hypodermis below the dermis, the two parts of the dermis in the middle, and the epidermis, the outermost layer of the skin. All of these parts have different types of tissue in them. So I'm going to type the name of a tissue in the chat. I'm going to say it, and then I'm going to type its name in the chat. I want you guys to tell me which part of the dermis it's found, or which part of the skin, excuse me, it's found in. So I already gave you a hint, right? So here's the type of tissue that I want you to tell me where I find it. This type of tissue is called dense, irregular, connective tissue. Dense, irregular, connective tissue. Which part of the skin? Okay, somebody try. Awesome. Somebody turned on their mic. Yeah, so dense, irregular connective tissue is found in the reticular layer of the dermis. So I'm going to add that. Dense, irregular, and I'm going to abbreviate connective tissue. So dense, irregular connective tissue is the kind that if you have watched any of the videos or have come to class with me, I've said this kind of tissue is really ugly. So I've put a picture in here that you've had in that PowerPoint with all kinds of tissue images in it. It's the messy kind of connective tissue that I find all throughout the dermis, the reticular layer of the dermis. The reticular layer of the dermis is the biggest part of the dermis, so there's a lot of dense, irregular connective tissue all throughout the skin. When I talk about it being dense, irregular connective tissue, its name gives me two important ideas that help me to identify it. The first part of its name is this part, this dense part. Whenever you see the word dense in the name of a connective tissue, that means I've got a lot of proteins in it. So when we look at this tissue, let me zoom in a little bit here. When I look at this tissue over here, notice that pretty much my entire space is filled with proteins. All of this pink stuff is proteins. Or sometimes you'll see it, it also kind of looks brown on some of our slides. So if I have an area that's completely filled with proteins like this, that's a dense connective tissue. When I say that it's a dense irregular connective tissue, that means that the proteins that fill up the space that make it dense are arranged irregularly, meaning some of them are running parallel to our board here. Some of them are sticking out of the board at you. Some of them are going at an angle. This irregular word means that they're not all going the same direction. They're going all kinds of different directions. That helps us, by the way, when we're thinking about the function of this type of tissue. What were some of the things that we said with our group is the function of dense, irregular connective tissue? What did we say about dense, irregular? Yeah, so specifically with dense irregular, our emphasis, I'm just going to type in here, multiple directions. Multiple directions. Yeah, it withstands pulling, or it provides strength in multiple directions. The reason this multiple directions is important is because that's why my proteins are going nuts. They're going all over the place. Because see, one of the biggest places we find dense irregular connective tissue is in this layer of the skin, this reticular layer of the dermis. Think about how when you're doing your everyday life, in your fingers, you move your fingers to type, you move your fingers to text, to do your hair, I don't know. You're constantly moving your fingers in all kinds of different directions. I need a type of tissue there that is going to make it strong. The connective tissue needs to be strong regardless of which way I'm moving my fingers. So dense irregular connective tissue is the kind of connective tissue that has a mess of proteins all over the place, going all kinds of directions. That's good for me because it helps me pulling in multiple directions. Now there's another type of tissue I'm going to mention that we don't have in the skin, but it's similar, at least in name, it's similar. The similar kind of tissue that we have to dense irregular is dense regular connective tissue. Can you help me out in the chat? Where would I find dense regular connective tissue? Where's dense regular found? Yeah, so a couple of us are chiming in. Dense regular connective tissue, the biggest place to know that one is that it's found inside tendons. And here's the thing, yeah, tendons and ligaments, to be fair, they both connect things together. Here's the thing with dense regular connective tissue. This is a kind of tissue that also has a bunch of proteins, that's what dense means, but these proteins are arranged regularly. They're all going the same direction. When you think about doing things like moving your entire arm or moving your entire leg, I have a fixed range of motions that I'm doing at that joint. I want to make sure that the movements or the directions I move things at that joint are really reinforced. So I put dense regular connective tissue at those joints because I'm always pulling in one direction. So the biggest difference between dense regular and dense irregular is when I talk about the strength of dense regular connective tissue, regular, we're going one way, I'm really strong one way. When I talk about dense irregular connective tissue, I'm really strong in a bunch of different ways. So the kind of dense connective tissue that I find in the reticular layer of the dermis is dense irregular connective tissue. And like we talked about, that's the one that gives us strength in multiple directions. Hey, I know I saw in the chat a question about fibroblasts. Let me add a quick note about those guys. Fibroblasts are a type of cell. Fibroblasts are cells that make fibers. Fibroblasts are cells that make fibers. When we talk about connective tissue, many types of connective tissue, basically any type of connective tissue that has collagen in it, has a lot of fibers in it, is going to have cells called fibroblasts. So you can't really see them when we're looking at dense irregular connective tissue because there's just so many proteins. But the type of cells that we would find in this kind of tissue, if we could see them, they would be called fibroblasts. They're cells that build, that's what blast means, builder, cells that build fibers, fibroblasts. When you're trying to identify dense regular connective tissue, you can see those fibroblasts a lot better. And we'll definitely look at some dense regular connective tissue so I can point out those fibroblasts to you. But that's the name of the type of cells in any kind of connective tissue that has these big collagen fibers in it. There's another type of tissue, let me zoom out here. There's another type of tissue on our big thing that we were working on here that has fibroblast cells in it as well. Can anyone tell me which part of the skin also has fibroblast cells? I actually asked you about these fibroblasts? Yeah, so it's this section up here is the other place in the skin that we find fibroblasts. So I'm going to add that little note right here, fibroblasts. Fibroblasts are the type of cells that we also find in the papillary layer of the dermis. Now, I know it's hard to see on my picture up here, the type of tissue, but I gave you an image that we've looked at before for areolar connective tissue. These fibroblasts, these fiber building cells, that's all the dark spots that you see when we look at this type of tissue here. Help me out in the chat really fast. What's the name of this type of tissue that I find in the papillary layer of the dermis? I want to ask you guys so I can start saying its name. What's the name of this tissue up here? Yep, absolutely right. Areolar loose connective tissue. Areolar loose connective tissue. We're going to abbreviate. Areolar loose connective tissue is the type of connective tissue that we see up here in the papillary layer of the dermis at the top of the dermis. This is a small portion of the dermis. Most of it is the reticular layer, little area up here at the top filled with this areolar loose connective tissue. Its name tells me that the protein fibers are loosely packed. That's why when I look at this kind of tissue, I can see some white space on my slide. I can see the little spots that are those fibroblasts, that are the cells. I can see kind of faint outlines of the proteins back behind them, but I don't see all of the dense proteins. Let me move back over really fast. All of those dense proteins like we saw right here. So this is a dense connective tissue because all parts of what I'm looking at are filled with proteins. That's what dense means. When I come over here, I see that there's still some open space in between those cells. That makes it a loose connective tissue. And areolar loose connective tissue is the type that we find in the papillary layer of the dermis. Miriam, I saw your hand go up. Did I answer your question or do you still have a question?
Sorry. Yeah.
You're good.
Yeah, my question was about the part that you actually just explained. Because as a reminder for myself, when I was studying tissues, I was connect the fiber with the dense. So in my head, fiber like those lines that will connect all the part of the tissue together. And I was thinking that the loose connective tissues are ones that without fibroblasts. But you said the fibroblast is kind of still there, but they kind of not tightly packed. So
yeah. So the fibroblasts are doing the same thing in dense and loose connective tissue, but the biggest difference between them is how many fibers they've made. So in areolar loose connective tissue, they haven't made as many protein fibers. That's why it looks like they're more loosely packed, where there's still space. When we do a dense connective tissue, we still have cells called fibroblasts. They're still building proteins, but they're building a ton of proteins. So there's not as much space for the cells because they're completely surrounded by proteins. Does that help?
Yes.
It is the same cells. They are doing the same job. It's just when we talk about areolar loose connective tissue, maybe you could think about those fibroblasts as being a little more lazy. They don't build as many protein fibers.
So I'm sorry.
Go for it. Yeah, you're good.
The main idea is the fibroblasts are there in any connective tissue, either it's loose or dense. It just the only difference is in the dense is like more active and more exist than the loose.
Yeah, basically.
Okay.
Almost all of my connective tissues do have fibroblasts. The exception would be this one down here that I asked you guys to identify. I don't have a lot of protein fibers down here in this place called the hypodermis. So the cells that live down here in the hypodermis, they're not called fibroblasts. We actually studied this in lecture. Can you guys help me out in the chat? What's the type of cells that I find in this kind of tissue?
Adipocytes.
Not fibroblasts anymore. Yeah, exactly right. The specific types of cells in the subcutaneous layer are called adipocytes. Adipocytes. The name of these cells matches up with the name of the tissue that I find in the subcutaneous layer or in the hypodermis. The type of tissue that I find down here is called adipose loose connective tissue. Adipose loose connective tissue. So if you recall from when we did our histology lab, the way that you identify adipose tissue, let me zoom in on it, is by these big, large open cells. If this tissue was alive, these big open spaces would be filled with lipids, or we would see the oil that's stained inside of them. Each of these cells are completely filled with the oil. So again, their name, scroll down a bit, is adipocytes, the special kind of cells that are found in adipose loose connective tissue. And a big location, one of the best locations for you to learn adipose loose connective tissue is in the hypodermis, or it's in the subcutaneous layer of the skin. So all of this down here, that's adipose connective tissue. Zoom back out. Two other types of tissue for us to identify. Let's do this one up here. When I'm talking about the epidermis, before you type its huge long name, because that's really long, which of the main types of tissue is in the epidermis? So main tissue types, that's things like connective tissue, muscle tissue, epithelial tissue, or nervous tissue. Let's start big picture. Which of those four types makes up the epidermis? Yeah, several of us are chiming in. The epidermis is made of epithelial tissue. When we talk about epithelial tissue, we can use the number of layers of cells that are present, and we can use the shape of the cells that are present to help us figure out what I would call these types of tissues. So when I look at the epidermis, I have more than one layer of cells. There's a layer of cells right here and some above it and here. We go all the way out. All of this is layers of cells. What is my word in epithelial tissue for more than one layer of cells? What word do I use to mean more than one layer? Yeah, a couple of us are chiming in here. The word for more than one layer is stratified. I'll type that on there for us. Stratified, more than one layer. Whenever, anytime we're looking at a stratified epithelium, we're looking at cells that have more than one layer. We always look at what's called the apical surface, or the top surface, to figure out the shape of our cells. The shape word that I'd use for these cells is the word that means they're really flat, or they're kind of squished. What's the name for cells that are really flat or kind of squished? Yep, coming in for me. So my word for my cells that are really squished or that are really flat, those cells are called squamous cells, or squamous, however you want to say it. Squamous cells. There's something special about these stratified squamous cells. These stratified squamous cells, when you look at them on the top, they are falling off. They are completely dead. It's because they're filled with a certain kind of protein. What kind of protein fills these cells out here? Or what's the last word that I need in their name that goes at the very beginning? Yep, so the protein's name is keratin, right? Keratin. That means that these cells are called keratinized stratified squamous epithelium. This tissue out here, keratinized stratified squamous, and let's add that last word here. See if I can fit it somewhere. Epithelium. Kind of fits. Here, let's do my mad magic up here. We'll move it up. Okay. Keratinized stratified squamous epithelium. That's the kind of tissue that I find in the epidermis layer of the skin. By the way, the epidermis is the only place that I find keratinized stratified squamous epithelium. When I'm talking about places called my mucous membranes, so the lining of the mouth, the lining of the vagina, these places also have stratified squamous epithelium, but it's called non-keratinized. It's not going to be falling off and flaky like this because it doesn't have keratin proteins. Those keratin proteins help the epidermis to do its particular job. Can you guys help me out in the chat? What was the job of the keratinized stratified squamous epithelium that I find in the epidermis, that I find on the very outside of your skin? What's its function? Absolutely. Yep, it's all about protection. So protection from abrasion, from UV. A couple of us mentioned water loss. Absolutely. Keratinized stratified squamous epithelium. This kind of tissue keeps you safe. This is your boundary between inside and outside. So I put those keratin proteins in it that helps to prevent me from things rubbing off and damaging my skin, keeps me safe from UV radiation, keeps my water inside. Hey, we're talking about this week in lecture, we're talking about the skin. That's our lecture topic. So we will talk even more about keratin and how that helps your skin. But the big thing you should think with keratinized stratified squamous epithelium is that this is a type of epithelial tissue that really helps to protect you, protection. My last one down here, my last picture down here, I didn't necessarily expect you to be able to identify it on your own. What I expected you to be able to do is use its location to help us predict what we would call this type of tissue down here. When I'm looking at this type of tissue, I am looking at something that is made all of cells. So I am looking at a type of epithelial tissue. When I look at this epithelial tissue, what would be the word I would use to describe how many layers of epithelial tissue we see here? Is this one a simple or a stratified? Yeah, so a lot of us are chiming in stratified. This is stratified. There is more than one layer. Now, it kind of looks like in this picture there's only maybe 2 layers. But remember that the word stratified just means there's more than one. So we've got a stratified epithelium here. When we look at a stratified epithelium, we always look at what we call the apical surface. When we're looking at stratified, well, we have it in the chat, so I'll mention it, stratified cuboidal epithelium, the place we find it is inside those sweat glands. They make sweat and dump it into those tubes that you guys saw on the skin model to bring that sweat all the way up to the surface of the skin. So the apical surface of this tissue right here is actually right along this open space here where it'd be dumping its sweat. When you look at these cells inside here, they are out as wide as they are tall. These are cuboidal cells. So the sweat glands that we find in the dermis area are called stratified cuboidal epithelium. Stratified cuboidal epithelium. Help me out in the chat. What did we say the function of this kind of tissue was? What's the function of stratified cuboidal epithelium? Yep, absolutely. Secretion. Secretion and sometimes absorption as well. But the big one that I think will be easiest for you to remember, since these are in sweat glands secrete sweat. They make sweat. So when you're thinking about the function of stratified cuboidal epithelium, remember that I find its location is in the sweat glands, and it secretes sweat. It will also secrete things like hormones if we are in a different area of the body. So stratified cuboidal epithelium in those sweat glands. Help me out in the chat. How do we feel about this activity now that we have worked on it together? Send me an emoji. Send me any questions that we still have. What do we think about this activity now that we've worked on it? Mike's got the scream going on, like that skeleton, right? That so many of us related to. Tyranny's feeling better. Awesome. Here, I got to send you guys my favorite emoji, right? Here comes the penguin. I'll try to draw you one too. Remind me at the end of class to draw you one. Christina asked, is it safe to say that cuboidal epithelium mostly secretes? Yeah, whether we're talking about simple cuboidal or stratified cuboidal, honestly, just remember secretion and absorption and you're good. Anytime we've got cuboidal epithelium, that's secretion and absorption, absolutely. So if you learn that function, you're set for both of them. So the question is, can we explain the difference between keratinized and non-keratinized stratified squamous epithelium? I can, but actually what I want to do is I want to ask my friends here in class, what are some things that you guys know about keratinized versus non-keratinized stratified squamous? What are some of our thoughts about the difference between them? Hard questions, right? Okay, yep, so Nicole mentioned that with keratinized cells, the cells on the top are dead. Absolutely. The first way you're gonna recognize them is you're gonna see that the cells are falling off on the top. That's definitely keratinized. If they're still attached, if they're still plump looking cells, we'll look at that non-keratinized today. But if you don't see cells that are falling off, then you are looking at non-keratinized. Keratin makes the cells stronger. Like Hunter mentioned, we find it in the epidermis. So really the biggest difference just comes down to, are the cells keratinized or are they not keratinized? Are they filled with this protein or are they missing that protein? If I fill the cells with keratin, they die, they get stringy and they start dropping off. If I don't fill them with keratin, they stay alive. A couple of people mentioned in the chat too, when we're looking at the pictures of keratinized versus non-keratinized, that non-keratinized tissue almost kind of looks thinner, or it looks shorter, if you will. There's not as many layers of cells. So that might be a good way to think of it too. But we will look at some pictures today. I'll show you non-keratinized versus keratinized. Biggest difference, the biggest difference for me, the way I identify it, is whether or not things are falling off. If they're falling off, we're keratinized. If they're not falling off, we're non-keratinized. You're welcome. Yep. And thanks to the class for helping us out there. Any other last minute questions about this before we move on? I will mention for you too, as maybe some of us are still typing, the more you can learn the locations of these types of tissues in the skin, the better off you are. Because think about it, we're kind of double dipping when we talk about the types of tissue in the skin, because we learned these tissues in lesson number 4 when we're doing histology. And then in lesson number 5, we talked about the parts of the skin that had those same kinds of tissues. So if I can learn what we just did on this slide here, if I can learn these as the locations of these tissues, that's one less piece of information that I have to learn otherwise, because their locations in the skin is going to be a huge help for you as your locations to learn. So make sure we know these as the locations of these types of tissues. Hunter is saying, is there a link where we can find images of the tissues? Okay, so I've got a couple of friends that say the link in the student Google Drive is not working. So my question for my friends that are having issues, can you get into the Google Drive at all, or are you not able to get into the Google Drive? That's my first question. Okay, so for any friends, for any friends who cannot get into the Google Drive at all, when you click on that link and it won't let you in, it's because you have a personal Gmail account, or somebody in your house has a personal Gmail account. You need to log completely out of that personal Gmail account. Make sure it's just your TCC one that's logged in. That will help you to get into the Google Drive. For those of us that are having trouble with the file, I would, like Christina suggested, I would say download it. It may not show you the file when you're online, because it is very large. There are lots of pictures. So try to download it onto your computer. I promise there's no viruses. So download that onto your computer, save it onto your computer, see if that works for you. And if not, I will be around. I'm going to have some open office hours tonight from 5 to 6:30. If you're still having trouble, plan to stop by tonight and we can try to troubleshoot it together. But hopefully the downloading thing will work for you. It may not work for you to view it online because it is really big. There's lots of pictures. So give that a try. Good question though. Yeah. And thank you to the class for our troubleshooting answers there. All right, here's the other thing that I had been hoping to have you guys do. We'll just do it together here as a class. Everyone's favorite, right? There's some more sarcasm. Dr. Ollis likes sarcasm. Everyone's favorite is identifying the phases of mitosis on slides. Before we identify these particular phases on my picture, yeah, some of us are crying. Before we identify these phases on our picture, can you guys list for me in the chat the names of the phases? You don't necessarily have to list them in order, although Tatiana helped us out there. She gave us a list for their names. Okay, perfect. Awesome. Yep, there. I think in our chat we have them all. So let me make this here on our slide here. So when you're thinking about the phases of mitosis, the way to remember what the parts of mitosis are is PMAT. So PMAT, we start with prophase, then we move into metaphase, then we move into anaphase, and finally we have telophase. Now, there are two other things that we talked about in relation to the cell cycle that were not part of mitosis that are important. Yeah, so my letters that Gloria mentioned for us are I and C. So one of my parts of the cell cycle that's not part of mitosis, but it is important for us to know, is called interphase. And then my last part is the thing called cytokinesis. I've said this before in our office hour sessions. If the wording on your question asks you, which phase of mitosis does something occur in? Your only options for phases of mitosis are these guys right here. If I ask you about the phase of mitosis, the only parts of mitosis, PMAT. It's got to be prophase, metaphase, anaphase, telophase. Got to be that. If I ask you, and here, let me type this terminology for you here. If I ask you for the part of the cell cycle, if I use this terminology right here, the part of the cell cycle that something's happening, I'm more likely to be asking you for interphase or cytokinesis. Cytokinesis technically means I am dividing. Kinesis means dividing. Cyto means the cytoplasm. Can you guys help me in the chat? What is the cytoplasm of a cell made out of? What's in the cytoplasm? Cytokinesis. It involves that cytoplasm. Yeah, so one of the big things, the thing we talked about in lecture that relates to the cytoplasm is the cytosol. It's the soup. The soupy stuff, and then also all the organelles that are living in the soupy stuff. When I divide the organelles and the soupy stuff, that's called cytokinesis. When I'm doing mitosis, what am I trying to divide in mitosis? What is mitosis all about dividing? Yeah, there we go. A couple of us are mentioning it. Mitosis equals DNA dividing. DNA dividing. So we got to divide the soup in cytokinesis. We divide the DNA in mitosis. So check that out when you're reading a question or when you're thinking about the difference between things. Are we talking about the cytosol and the organelles getting split up, or are we talking about the DNA getting split up? If I say the part of the cell cycle, I'm probably talking about dividing up that soup. If I say the phase of mitosis, we're talking about that DNA. When we talk about interphase in the cell cycle, what are some things we know about interphase, that part of the cell cycle? What's going on in interphase? Yeah, so a couple of us have mentioned this is when a cell grows. Absolutely. This is when it grows. This is when it's replicating DNA, when it's making a copy of its genetic information. Gloria made a good point too. Most of a cell's life cycle is spent in interphase. Most of the time it's growing, then it copies its DNA, then it grows some more, then it goes through the process of mitosis, where it divides up its genetic information, and the process of cytokinesis, where it divides up its soup. So, important thing to remember. I a lot of times will say in our sessions together, underline, highlight, star, idea, meaning this is important. If you see this terminology right here, the part of the cell cycle that something's happening in, that is going to be either interphase or cytokinesis. If I ask you for the phase of mitosis, now we're talking about these guys here. Now we're talking about PMAT. So watch that terminology. That will help you know what we're looking for. For each of the cells that I have tagged here with arrows, the question would ask you about the phase of mitosis that my cells are in. So here, let's do this as a class together. So I'm going to clear out my board. We're going to start over. If you see, so I'm going to type in my phases here. If you see a cell that is in prophase, in a moment, I'm going to have you circle that cell in red. In a moment, please, not yet. If we see a cell in metaphase, we're going to circle that cell in blue. If we see anaphase, we're going to do green. And if we see telophase, let's do black. Telophase. Okay. In a moment here, we're going to go nuts, and my screen is going to look crazy in the best possible way. Up above this pointer, see if I can get this to go away. Up above this pointer right here, there's a pencil. When you click on that pencil, then you can go over to the side, and there's a circle that has colors in it. Pick the color that represents the phase you see. Pick that color and then go through and circle the cells in those phases. So everybody go ahead and grab your pencil. It's completely anonymous. Pick your correct color, and then let's go circle some cells that we see in these phases of mitosis. Let's see if we can find our phases of mitosis. I like it. Look at how beautiful our artwork is. Okay, so it looks like we mostly have consensus here. And I know a lot of us focused on the cells with the arrows, right? So we mostly focused on the cells with the arrows. But props to my couple of friends down here who recognized I had another cell that we could identify. So to help us be able to see what's underneath all of our beautiful drawings here, I'm going to go ahead and clear it off. Say goodbye to our little smiley face. Hello to my friend who's happy enough to draw a smiley face, right? All right, I'm going to clear it, and then we're going to look at it together. And a heart. Look at that. Wonderful. All right, here we go. We seemed to agree as a class that when we're looking at, let me get my pointer back, this cell right here, remind me in the chat, which phase of mitosis did we say that this cell was in right here? Which phase of mitosis was this one? Yeah, so we identified when we were looking at this cell that this cell is in prophase. So some reminders for us here, I'll type it on our chart. This is a cell that is in prophase. when we talk about the phase of mitosis called prophase, what are some of the most important things that happen in prophase? What does a cell do in prophase? Yeah, so the first thing that happens that Nicole mentioned for us is that the DNA condenses, or maybe to put that in easier words, what we could say is that chromosomes form. I have to move this. There we go. Chromosomes form. So normally in your cell, we have that soupy stuff, or excuse me, we have the stringy form of DNA that's called chromatin. That's a review word for us here. I'm going to give it to you at the bottom. Chromatin. This is when your DNA looks like a wet spaghetti noodle and it's chilling out in the nucleus. Chromatin is really great for a cell to do the process of transcription and translation, which is what we talked about in lecture last week, right? So chromatin, great for transcription and translation. Chromatin, not great for dividing into new cells. So we form chromosomes, which are those X-shaped forms of DNA. We form chromosomes when we are in the phase of mitosis called prophase. So the first thing that happens in prophase is I make chromosomes. What happens toward the very end of prophase? Does anyone know what I'm thinking of here? It actually has happened on this particular cell. There's something else, not the mitotic spindle that I'm looking for here. There's something that disappears or dissolves. What do I lose at the end of prophase? Do we remember? Yeah, the nuclear envelope. Yep, so nuclear envelope, or sometimes it's called the nuclear membrane. The nuclear envelope disappears. That's the second thing that happens in prophase. And this is something that helps us when we're trying to figure out if a cell is an interphase or prophase. If I don't have a condensed or a confined space where my genetic information is, and I can see this little dotty or kind of squiggly looking genetic information, that cell is in prophase. So as we can see our cell right here, I've got little dots. That's the condensed genetic information. It's not together in one big circle. That one big circle would mean that I still have a nuclear envelope. I know it's kind of chopped off here, but if you notice down here how all of my genetic information on this one is all together in one enclosed space, as opposed to kind of spreading out. This one down here is probably a cell in interphase, where its genetic information is all still together in the nucleus. It's loose, it's in the form of chromatin, and I have a nuclear envelope holding it in place. So the two things you're looking for to identify prophase is we're looking for DNA that's condensed down. It's going to look like little polka dots or little strings. And we're also going to see that the nuclear envelope is missing. That DNA is kind of floating around, free floating as a mess. This is a really good prophase cell. We had two cells, this one and this one right here, that were in the same phase. What phase are these two cells in right here? What's the phase on these ones? Yeah, these ones are in metaphase. Metaphase. The way that I like to remember metaphase is that the chromosomes meet in the middle of the cell. When we talk about metaphase, there's a structure that we can see called the metaphase plate. The metaphase plate, that's this line right here, the metaphase plate. And that's why I can see that line right here too, the metaphase plate. When a cell is dividing its genetic information, it basically plays tug of war. So I've got two centrioles, one on each side over here. They're tugging on those chromosomes. They tug back and forth, back and forth, back and forth until those chromosomes end up in the very middle. Eventually what's going to happen is we're going to pull so much that the chromosomes are going to split. They're going to break in half. But metaphase is the stage where they all line up in the middle of the cell. They meet in the middle of the cell to form this line. So the biggest way to recognize metaphase is when those chromosomes are in a line, that metaphase plate. What's going on in this cell over here? What phase did we say this one was over here? Do we remember? Yeah, so this one over here is an example of anaphase. This one is anaphase. A couple of ways I can tell this one's anaphase. First way I can tell is if you notice I've got 1 and 2 groups of genetic information. Those sets of genetic information, see how the lines are kind of pointing toward the middle of the cell that's dividing? What's happening here is my chromosomes, let's do a little freehand here. My chromosomes started as an X, and I used my mitotic spindle to connect to each side of them. I'm playing tug of war with them in metaphase. Well, after metaphase, I enter anaphase, and what's going on there is half of my X is getting pulled toward one side, half of my X is getting pulled toward the other. So if we were looking at this when the cell was alive, what would be happening is these two black lines are getting pulled in opposite directions. So you see that the DNA kind of trails away from the middle of the cell because I've chopped my chromosome in half and I'm dragging it away. So I'm dragging it toward the two ends of the cell. So in anaphase, you're going to see two distinct areas of DNA, but that DNA is still going to look stringy. It's still going to kind of be in a line, and you're going to see that it's trailing toward the middle of the cell. Yeah, so Mike asked in the chat, in anaphase, the DNA is always splitting. That's correct. Yep, anaphase means we've broken up the chromosome. We're pulling that information away from each other. The last phase of mitosis, I don't really have on this picture here, that last phase of mitosis. The last phase of mitosis is called telophase. Telophase. And the big thing with telophase is when it happens, typically what's also happening at the same time is cytokinesis. So typically, telophase happens at the same time as cytokinesis. Telophase, when I ask about the phase of mitosis, what I want you to focus on is what's going on with the DNA in a cell. When I get to telophase, my DNA is all the way divided. So these chromosomes that I was pulling apart in anaphase, they're all the way pulled apart in telophase. And when we get to telophase, a couple of things happen. We form two new nuclei. So two new nuclear envelopes to cover up my DNA. I put it in its own little new nucleus. The other thing that happens is the DNA relaxes, or we make chromatin again. In telophase, since I have new nuclei, I've got a new nuclear envelope, I'm going to relax my DNA. It doesn't need to be in that X pattern anymore. Now I can relax it, and it can be in two new nuclei. Now, the other way, just practically speaking, the other way you will identify telophase on a slide is you will actually see, yeah, like Gloria mentioned, we will see evidence of cytokinesis happening. We're going to see the thing called the cleavage furrow. So let me add that to our slide here. We're going to see the cleavage furrow form. So if I was looking at this particular, so let's go back here. If we were looking at this one and this one was in telophase, we would see a couple of things that would look a little different. The first thing we would see is that cleavage furrow. So what that means is there's an indentation in the middle of the cell. If we looked a little bit more like this, that would be a cell that is in telophase, because I can see that cleavage furrow developing. The other thing we would see on this slide is if my DNA, instead of being pulled away, if my DNA was more of like a black spot, I've got two dark spots here that are fuzzy. Instead of having these little stringies, if I had two separate spots where we're not pulling the DNA anymore, this cell would have been in telophase. But because those chromosomes are still moving, they're splitting from each other, this is a cell that's in anaphase, not telophase. Telophase would look a little bit more like this. What questions do we still have about mitosis? Or give me an emoji. What do we think about mitosis? Tatiana has a little stick to her stomach. I feel ya. Let me remind you, I'm going to pop a website into the chat right here. If you remember back from our live when we talked about microscopy, I told you guys about that virtual microscope. That is the link to the virtual microscope. So let me put it on the slide for my friends that are joining via recording. There's the link to the virtual microscope. When you are in that virtual microscope, in the slide box, they have, it's listed as animal slides. So let me add my directions here. If you click on it that says animal slides instead of human slides, they have the phases of mitosis. They have slides that when you get all the way up to 40 times your high power objective, or if you want to go all the way up to higher than we can go in our lab to 100 times, you can see the individual phases of mitosis in images. So check out the virtual microscope. Again, go to that slide box and find the animal slides, and then there's going to be things like late prophase or metaphase or late anaphase. You can go through and see specific examples of these phases of mitosis. So another study tool to help you out with mitosis. Yeah, so when you're working on identifying these phases and on everything on the exam, yes, the format is going to be the same as what you're used to on homework assignments. So the drop-down menus, the multiple choice, there might be some that are multiple answer, where we ask you to select all of the types of a tissue, or select all the statements that are true, that kind of stuff. It's going to be the same format as those homework assignments. So let me pop up really fast. We will do this in a moment. I want to, since we just talked about mitosis, let me go to my mitosis slide right here. When we look at our mitosis slide here, help me out. This one right here. Can we tell what phase we got going on in this picture up here? Yeah, this one up here is a telophase cell. Two ways I can tell. My DNA has clumped together. There's no more stringies toward the middle, and I can see that cleavage furrow. So this is a really great example of a cell that's in telophase. I can see that cleavage furrow, and I can see those two distinct sections of DNA. Notice right here how my DNA is still splitting. I don't see a cleavage furrow right here. Yeah, so this one right here that I'm looking at, this is an anaphase cell. So if you compare that DNA being dragged apart to the DNA that's completely separated, that's the big difference between anaphase and telophase. What do I have going on right here? What's this one right here? Yep, there's that metaphase plate, right? That line of cells. They're meeting in the middle, so there's that metaphase plate. Hey, check out these cells right here. These cells right here have DNA that has condensed. Which phase of mitosis does the DNA condense in? Yeah, so these guys are prophase cells. DNA condenses. We can have prophase that happens before the nuclear envelope dissolves. So see how when I'm looking at this one, I can see that DNA in particular places. Notice how with this one, the nucleus on this cell is pretty much all dark. This nucleus being pretty much all dark compared to, let's compare it to its neighbor. See how there's some spaces up here where inside that nucleus is a little bit lighter? Prophase is when I start to see some distinct lines compared to the fact that this entire nucleus is pretty much full of DNA. So prophase, the best way to identify it is either like that one that we looked at, where there's no nuclear envelope left, or if you can see very distinct clumps of genetic information inside that cell as opposed to just fuzzies, as opposed to just this mess that we see up here. The difference between prophase and interphase is having condensed chromosomes, seeing that genetic information. So try to use your super sleuthing skills to tell the difference between those. Here's the other thing I'm going to say right here. Check out your wording. phase of mitosis versus part of the cell, I can't spell this morning, sorry, cell cycle. If I ask you a question on the slide and I tell you to tell me which part of the cell cycle this cell is in, which answer you're going to give me? Which part of the cell cycle would I say that this cell is in? Part of the cell cycle. Yep. Interphase, right? Part of the cell cycle, my options are cytokinesis or interphase. If I ask you about a cell and I say, which phase of mitosis is that cell in? What are we going to say to that? What's the phase of mitosis? Prophase. Yep, prophase. So check your terminology. There's your other tip off. We're looking to see what that genetic information looks like, whether it's a little bit more condensed or whether it's looser inside the nucleus. And we're looking to see the way that the question is worded. Am I asking you about mitosis or am I asking you about the cell cycle? So check for those hints when we're trying to decide on prophase versus interphase. Tatiana, I am not sure if we'll be able to zoom in or not. I will try to find you an answer for that. I would say to be safe, assume that you can't, because I would hate for Proctorio to kick you out because you tried to zoom in. But feel free to go up as close as you want to your screen to look at stuff. I believe the pictures are pretty big, hopefully. So, but yeah, just to be safe, I'd say probably don't try it, and I'll see if I can get some clarity on that. I'll mention this too when we're on the topic of Proctorio. If for some reason when you're taking your exam today or tomorrow, if you for some reason get kicked out, like your internet dies or something pops up on your screen and it ends your test for you or something like that, please get in touch with Proctorio first. I cannot get you back into that exam. Proctorio can get you back into that exam. So I put a link on the announcement that I posted for you guys last night. If you have any trouble with your exam kicking you out, go there first and start a live chat with Proctorio. They're really good about getting students back into the exam. But if for some reason they can't help you out, then get in touch with me. But I will not be able to get you back in the same way that they can. Oh yeah, so Gloria is saying for MacBook friends, make sure we're in Do Not Disturb. Absolutely, yes. I had a couple of students that had issues with that. So if you have a MacBook, make sure you're not going to get any kind of notifications, because Proctorio says that that's not going to fly and it's going to literally kick you out of the test. So MacBook friends, do not disturb. I don't have a Mac. Can someone help out, Jacqueline? How do we get our Macs into do not disturb? It's a great question I can't answer. Okay. Perfect. Lauren says she'll snap that over. Hey, and maybe do me a favor. If we can send out a quick note to our GroupMe chat for our class about that, that will save a lot of friends time and suffering. So if you are in your GroupMes, if somebody can just send out a quick message to everyone to remind them if they have a MacBook to go and do not disturb, that will save much pain and suffering, I bet, for our friends. Okay, yep, and Gloria's saying check that storage to make sure that things aren't going to pop up. Yeah, and again, if you have pop-up issues that kicks you out of Proctorio, Proctorio is pretty good about getting you back in. So please contact them if you have some issues. All right, I have half an hour to address the other questions that we have. Let me get us a blank whiteboard here because that might be the easiest way. I would like for you guys to help me out on the whiteboard. I want you to type for me some of the things that you're interested in covering. What I will tell you is I had someone that asked me to cover the degrees of burns. So let me add that, degrees of burns. We wanted to talk a little bit about that. I've added that topic to our whiteboard here. If you have other things that you're interested in us talking about, can you go ahead and add them for me really fast? Hopefully we don't end up typing on each other. Okay, I've got a question about bone markings. See if we can make a quick list of what we want to see. Yeah, Jacqueline mentioned that the big red sign that says no access, if that pops up for you, that means that your proctorio is not working. So use that secure exam proctor. When you click on that, it will reactivate proctorio. That should get you into the exam. There is also a troubleshooting document that's posted in the lecture area of our class. So consider checking that. Okay, when it kicked you out, that screen. Yeah, so that is when you go and talk to proctorio, because they can hopefully get you back in. Hopefully we don't have that issue this time, right? Hopefully this test just goes smoothly. I'll mention too, for you guys, I have open office hours today from 5 to 6:30 and tomorrow from 12 to 1:30. Those are times when you know that I will be at my computer waiting to help you out. So translation being, that might be a good time for you to consider taking the lab exam, because I will be at my computer right away if you have issues. So today from 5 to 6:30, tomorrow from 12 to 1:30, I am 100% available right away. Mike, I'm not hosting a session like this one, but if you have particular questions that you want to bring to my office hours tonight, I'd be happy to chat with those as well. I mean, if we have a bunch of people that show up today, sure, I can do another review session. We can talk about some more stuff. So if a lot of people come, we'll do a review session. If it's just like 1 or 2 people, we'll have a chat. Okay, so let me check out my my list up here. So question about knowing the bones and the regions. We need to be able to identify the bones, absolutely. So there's going to be a couple, at least probably 2 or 3 questions about bone names. Make sure we know the bone names. Same deal with those regional terms. The regional terms are very strongly related to the bones. So these things kind of go together, and yes, we do need to make sure we know them. Friendly reminder, there's that midterm review assignment. If you want to go through and just do the bones questions and just do the regional terms questions, go for it, to help you practice those particular things. But yeah, make sure we know the bones, make sure we know the regional terms. For these questions here with the bone markings, we'll mention for you is that we will not ask you specific bone markings from the femur or specific bone markings from the skull. So what that means is, please do not memorize the location of all of those bone markings on the femur and memorize the location of all of those bone markings on the skull. What we do need to memorize is that list on the last page of the lesson number 6 packet, where we sort things into indentations, where we sort them into holes, when we sort them into projections. I actually covered this in depth in office hours last week. So I'm going to refer you to the video from last week. It was the supplemental video from one of our office hours last time, because we did group work and then we came back together to talk about these things here. So I probably won't have time in our class today to talk about this, but we did talk about it live last week. So check that out. If it's not helpful for you, that video, come back tonight and we can talk about this together. Yeah, we got our mitosis here, right? With a smiley face. So hopefully our talk today helped. I've got a lot of requests here with skin stuff, right? So the strata of the epidermis, probably some of those structures. And yes, I know we want to do the burns, right? Burns relate to my skin too. So it looks like we mostly want to dive into the skin. Can you guys help me out in the chat? We had a question about the differences between the functions of spongy bone and compact bone. When you guys are studying spongy bone and compact bone, what are some of the things you're learning for functions or ways that you're thinking about that? I want to put that question to the class. Spongy bone versus compact bone. Okay, yep, so compact bone is that kind that's found on the outside. Absolutely, spongy bone is the bone on the inside. When we're thinking about functions, yeah, the functions are similar to one another, but the big difference when we're thinking about those types of tissue is the bone marrow. Absolutely. So spongy bone is like a sponge. It has space in it. Because spongy bone has space, that's going to be the type of bone tissue that helps me to store bone marrow. There's no space to store bone marrow in compact bone. So compact bone is the strong kind of bone. That's going to be the type of bone primarily that is helping us with movement, that's helping bone to be strong or very supportive. Bone marrow is only stored in spongy bone. Nicholas, is everything else the same? So both compact bone and spongy bone have the same kinds of cells. So here's another cell review for us. Let me get my typing out here. My type of cells that I find in compact bone and spongy bone are the osteocytes. Both types of tissue have those bone cells. Next week in lecture, we're going to talk about the stuff that's outside those osteocytes. It's called, well, so its chemical formula is calcium phosphate. Both of them have calcium on the outside. We store calcium in our bones. So what's the same about them is their type of cells and the stuff in them that makes them hard. What's different is that spongy bone has space to store bone marrow. Compact bone does not have space to store bone marrow. So when you think about their functions, in general, bone tissue is what makes your body have a skeleton, what gives it support or protection. But spongy bone in particular also is the location of the bone marrow. Do we feel good about that? Thumbs up. Miriam has a question. Go for it.
Okay, sorry. My question is, when I was reviewing the recording for the bone slides, the slides that's on the lecture are kind of different, where the location of the bone marrow and the trabeculae. So I'm not sure if it's going to be time that we're going to look at this right now, but the main question is the bone marrow. My understanding is that little white part of the spongy bone, and the trabeculae is the surrounding area, right?
Yeah, so here is a slide that has a bunch of different types of tissue on it. One of the pictures on this slide is spongy bone. I'll put this to the chat. So Miriam, you're not on the spot here. For my friends in the chat, what letter represents spongy bone tissue? Can we tell? Yeah, spongy bone is F. Okay, so here's my spongy bone tissue down here, right? This is a picture of spongy bone. The two things we had to recognize on spongy bone tissue, we had to recognize things that are called trabeculae, trabeculae, and the other thing we have to identify is our bone marrow. When I look at spongy bone tissue, the trabeculae are these fingers that I see right here, so the solid parts. So I'll kind of trace on the outside here. All the stuff that's inside my black lines here, these are the trabeculae, the solid pink stuff. These places that I see right here, where I've got this red stuff in between the solid pink, or when you're looking at our other kind of spongy bone slide, you might see some purple or blue spots inside here. That is the bone marrow. So the parts of your view that are completely filled, that's the trabeculae, that's the bone tissue itself. parts that are a little bit more open, like we see here, that's going to be the bone marrow. So I see the bone marrow, and then I see the solid trabeculae, the solid bone tissue that's inside of it. Does that help, Miriam, in thinking of the differences between them? Good. Okay. Here's what I'll say. I will tonight, if a bunch of you guys want to show up tonight at 5, we can do tissues to death again. So if you guys want to do tissue practice tonight at 5, I will be here. So we might as well just call it a tissue prep night. So we'll go through and do things like this one here. We'll go through and talk about each of these types of tissues. We'll practice identifying stuff. So if you guys want to be here tonight at 5, I will be here. And yes, Tatiana, I will record it. So it'll be posted for you later this evening. So yeah, we'll plan to do some histology review together. So let me do skin, because I know I had some questions about skin, and then we'll do that tissue stuff tonight. So let me pull up, oh, right here. Okay, I'm gonna go down to the skin. Okay, when we talk about the skin, the good news is we've already talked about those regions of the skin already, because we did that labeling activity together. So I'm going to put some brackets on here. First thing we're going to do when we look at our big skin model here is we are going to label our regions of the skin. So I'm going to put some numbers so that I can type instead of having to go through and write everything in the right place. All right, so help me out in the chat. Region number 1 of the skin. What do I call part number 1 in the skin? What's the outermost region called? The outermost region is the epidermis. So region number 1, epidermis. Okay. Region number 2 is what part of the skin? That uppermost part there, that's the papillary layer of the dermis. Yep. Okay. Region number 3 is the really big one. Region number 3 is called the reticular layer of the dermis. Absolutely. Okay. And then region number 4 is that lowest layer. Had a couple of different names for it. I like hypodermis best. You might also see it called the subcutaneous layer. Add that word layer. Subcutaneous, by the way, also just means under the skin, subcutaneous. If you've ever heard about or had a subcutaneous injection, it just goes into this hypodermis layer, subcutaneous layer. Okay, we named these areas of the skin because that's going to help us out when we start talking about burns. So when we talk about burns, we've got 1st degree, 2nd degree, and 3rd degree burns. So 1st degree burns, not a trick question. 1st degree burn, how many layers of the skin does a 1st degree burn damage? Yeah, 1st degree burn is going to damage 1 layer of the skin. So 1 layer of the skin, it's going to damage the outermost layer of the skin. So when I talk about a 1st degree burn, the only thing that's going to get damaged is this outermost layer of the skin. I know this is going to be a long typing, so you can abbreviate. Remind me the name of the type of tissue out here. Which kind of tissue is in the epidermis? It's a super long name. There we go. Yep. So the epidermis, remember, keratinized, stratified squamous epithelium. Cells that are filled with keratin, lots of layers of them, and it's epithelial tissue. So all of this stuff out here is keratinized, stratified squamous epithelium. Here's one other cell type name. So when I have keratinized, stratified squamous epithelium, here's the name of that tissue type coming at you. The name of the special kind of cells in that type of tissue are called keratinocytes. Keratinocytes, the cells that live in keratinized, stratified squamous epithelium. All of this tissue out here is made of cells, and those cells are called keratinocytes. In a 1st degree burn, let's draw ourselves a little box here. 1st degree burn, I'm damaging this first layer of the skin right here. So I'm damaging my cells called keratinocytes. I'm damaging my keratinized stratified squamous epithelium. The other thing I might damage as well is this part of my hair. What's the name of the part of a hair that sticks out above the surface of the skin? What's that called? Yeah, that's the shaft of the hair. The shaft of the hair is everything that sticks out above the surface of the skin. My parts of the hair that are embedded in the skin, what part of the hair is this part called? What's this part down here? Yeah, this is the root of the hair. So the root of the hair is inside the skin. The shaft of the hair is outside the skin. In a first-degree burn, we're going to damage my epidermis layer with its cells, with its type of tissue, with the shaft of the hair here. Nicole asked if we would say that Apocrine glands are damaged. Because the part of the gland that actually makes the sweat is not found in the epidermis, we would say no on that. Their ducts stick up through there, but the glands themselves are actually okay. So we would just say that it's that tissue out here. We'd say it's those cells. We'd say it's the shaft of the hair damaged in a first-degree burn. In a second-degree burn, how many layers of the skin do we damage? Second-degree burn. Yeah, now we're up to 2, right? Okay, so in a second-degree burn, we still damage the epidermis out here, and our damage at least extends into the papillary layer of the dermis. So we're definitely going to damage the papillary layer of the dermis up here. We might go even up to about halfway through my, my reticular layer of the dermis as well. So let me add a box for us here. When we are talking about a second-degree burn, second-degree burns are going to extend down into the reticular layer of the dermis. So my green box is the second-degree burn. So in a second-degree burn, we're going to damage all this stuff on the outside. So those keratinocytes, that keratinized stratified squamous epithelium. But we're also going to start to damage some of the things that we see inside here. So help me out in the chat. Let's make some lines here. This structure right here that I got an arrow pointing to, it's something that's attached to the root of my hair. It's a type of gland. Does anyone know what kind of gland we're looking at right here that dumps its secretions onto hairs? Yeah, this is called a sebaceous gland. Makes sebum. So sebaceous glands, type their name on here, sebaceous glands. A sebaceous gland attached to a hair makes sebum. That's what is the fancy name for oil. Oil keeps your skin and your hair soft, makes them not brittle. So sebaceous glands, they would get damaged in a second-degree burn because they're found here in the upper part of the dermis. Let's talk about this guy. Oh, yeah, I'm in black. Okay, this guy right here that is attached to a hair. What is this little guy right here called? Does anybody know? Yeah, so this is the one muscle that we're learning with the skin. This muscle is called the erector pili muscle. Erector pili muscle. The short description of what erector pili does is it gives you goosebumps. Your goosebump muscle that I see right here is attached to the root of your hairs, makes them stand up when you have goosebumps. Since that's found here in this upper portion of the dermis, in the top half of the dermis, that would be damaged in a second-degree burn. These little guys right here are touch receptors. Does anyone remember the name of the light touch receptors? Yeah, so a couple of us are chiming in here. These ones that are found up here at the top, that's gray. These guys are called tactile corpuscles. Tactile corpuscles. I know this corpuscle word is weird. Just think nervous system. Whenever you see corpuscle, just think nervous system. Tactile corpuscles, they help me to do light touch. So when you're reading Braille, for example, light touch, discriminative touch, that's the job of these guys right here, tactile corpuscles. So first-degree burn just did the epidermis. Second-degree burn goes down into the dermis. By the way, how do you know that you have a second-degree burn and not just a first-degree burn? What's the big tip-off that I've got a second-degree burn? Yeah, it's those blisters. Whenever we see a blister, we've got a second-degree burn. I mentioned this when we were doing this together in class. The reason that you form a blister is because your UV rays come in and they go all the way through the epidermis and they damage the connections between the dermis layer and the epidermis layer above it. They basically fry them. And what happens is the epidermis layer separates from the dermis layer of the skin, and that leaves a space for fluid to rush in between. So a second-degree burn helps us to know I'm going to know I have it because I've separated my epidermis from my dermis. I had to go all the way into the dermis to get this blister to form. That only happens with a second-degree burn. Nicole asked about apocrine glands and a second-degree burn. So this particular picture doesn't have apocrine glands on it. Let me put this question to the class. Where do I find apocrine glands? Where are apocrine glands found in the body? Well, so I'm not asking skin region. Actually, I want more general than that, actually. What parts of your body have apocrine glands? Are they everywhere? Are they just certain places? Yeah, Vanessa's hesitant, but she's right. The only place in the body where we have apocrine glands, let me put a note about this down here. Apocrine glands, those are the really big ones that are on your labeling picture. Apocrine, cannot spell, sorry. Apocrine glands. I find those in axillary skin. That's the armpit, skin of the armpit. So to your question, Nicole, we could damage apocrine glands in a second-degree burn, but I think, can we all agree as a class, OMG, if I got a second-degree burn in my armpit, like, what in the world, right? What was I doing? I must have fallen asleep when I was tanning, and that would be so painful. You're absolutely right. If I were to somehow second-degree burn my axillary skin, if I were to damage down into the dermis layer in the axillary region, I would damage those glands too. So if that showed up on the test, we would say yes. Yeah. I don't think it is on the test, but if it is, we would say yes for a second-degree burn. Again, that just sounds painful. I am a very white person. I don't have a lot of melanin. We're talking about melanin this week in lecture. So I've done my fair share of sunburns, but I have not sunburned my armpits. I can say I have at least not done that. Jacqueline, lesson number 7 for lab is next week. We don't have new lab content this week. We're just doing new lecture content this week. My caveat to that, though, is the pace of content is going to start to pick up. I know that lab's already been crazy, right? It's about to start getting crazier because we're going to start learning bone markings and muscles and new content all at the same time each week. So when you finish with the lab midterm exam, I would recommend, here's your teacher's recommendation, start working ahead. So you finish that lab exam tomorrow, go ahead and start working on lab lesson number 7 on Wednesday, Thursday, Friday. Start working on it because it's a lot, especially when we get into, I think it's like week 8 and maybe week 10 or 11. The weeks that we do the arms and the legs are a lot to learn in a week. So if you can start working ahead this week, please do. You will thank yourself later as we keep moving on. Yeah, we got this. We're gonna rock this test. We're gonna keep going strong through this semester. But again, if you can work ahead this week, even though you don't have to do lab lesson number 7, I would say maybe consider it. Yeah, that's fitting, Ariel, that you put the flexing arm because we're starting to learn about muscle tissue in lesson number 7. So lab lesson number 7, muscle tissue. All right, the one other kind of burn I haven't mentioned, so we'll add a box for it here, is my 3rd-degree burn. 3rd-degree burn, I've got lit up here in blue. When we talk about a 3rd-degree burn, they call a 3rd-degree burn a full thickness burn. 3rd-degree burn is going to burn everything that we have in the skin. So all this stuff that we've already talked about here in the dermis. In addition to that, we're going to add in some of these guys that live down here. So way down here, let me get my pencil back. Way down here, we have a corpuscle, a nervous tissue structure, that helps me to detect pressure. What was the name of this guy that lives down deep? Yeah, so this guy that lives down deep is called a lamellar corpuscle. Lamellar corpuscle. These guys are found either in the very bottom of the reticular layer of the dermis or even down into the hypodermis layer. Because lamellar corpuscles are found so deep in the dermis, they are only going to be damaged in a 3rd degree burn. 3rd degree burns, called full thickness burns, we're going to damage all of the epidermis. In fact, we're probably going to burn it away. It's not going to be there anymore. We're going to damage pretty bad this papillary layer of the dermis. That might be gone. There's going to be a lot of damage here to the reticular layer of the dermis, and that might extend down into the hypodermis layer. So a 3rd degree burn goes through all the parts of the skin. It's going to damage everything that the other types of burns damaged, along with these lamellar corpuscles that are down deeper, along with my adipose loose connective tissue that I find in the hypodermis layer, along with those cells that we talked about called adipocytes. So 3rd degree burn, we burn it all. Everything that's in the skin is going to be damaged in a 3rd degree burn. How do we feel about burns and the skin? Thumbs up, thumbs down. What questions do we still have about the skin? Yeah, Miriam has a question. Go for it.
I apologize for the many questions that I have today.
No, you're good.
One of the questions on the midterm practice exam, they had, if you allow me, I'm going to draw an arrow here.
Okay. Here, actually, before you do that, let me clear off my board so that I can see for sure where you put it. So I'm going to clear it off. Okay, go ahead.
Okay, thank you. So on the very bottom, when they asked us to label some of the parts, if I can actually, I don't know how to get the arrow. Okay. Well, at the very bottom.
So if you go above here, see the little pencil up there? If you click on that pencil, it'll let you draw.
Oh, okay. It's not.
Oh, okay. Just describe to me what it looks like, and we'll find it together.
Okay, at the very bottom, yes, at the very bottom.
Right here?
Really like a little down. So the arrow is pointing at the blood vessel right here, but it's not part. Yeah, it's not part of the options. And when I thought about it, it might be pointing at the whole part of the dermis, which is the hypodermis. But on the side, they had another bracket pointing at the hypodermis.
Okay, so Jacqueline's mentioning, a couple of students are mentioning that yeah, it's pointing, so the arrow might be poorly placed. What it's actually asking you to look for is this Apocrine gland here, as opposed to the blood vessels there. It's asking about this particular gland. Oh, so it's probably just a poorly placed arrow. Sorry about that.
No, that's okay. That makes sense.
Yeah, okay. So instead of it pointing directly at the blood vessels, it's trying to get your attention here, gland that's right next to them.
Okay, that makes sense. Thank you so much.
Yeah, you're welcome. And I'll mention too, when we're talking about the difference between, we've got sebaceous glands, got apocrine glands, and we've got eccrine glands. When you're talking about the difference between them, check out where they're dumping their secretions. So this one right here, and its neighbor right here, and I got another one right here. They go all the way up to the surface of the skin. My eccrine glands are the ones that dump onto the surface of the skin directly. When we talk about sebaceous glands, the ones that make oil, they're always attached to hairs. So if I'm pointing to something attached to a hair, that's a sebaceous gland. If I'm pointing to something that goes all the way to the surface of the skin by itself, that's an eccrine gland. And the apocrine glands, they're not shown on this one right here, but the apocrine glands are really, really large. Only in axillary skin, very large. So to help you distinguish between them, keep those things in mind. Eccrine goes to the surface, apocrine, really big, sebaceous, attached to hair. Got a comment that says, with all the training in elementary school about stop, drop, and roll, am I the only one that thought catching on fire was very likely? Yeah, you know, we so teach children about stop, drop, and rolling, right? I guess we just want to make sure we don't get those third-degree burns. If we were in a chemistry lab, we'd have to worry about stop, drop, and roll a little bit more, right? There's not enough fire in our lives, obviously. Okay, so let's wrap up for this morning's session. We talked about the structures of the skin. We talked about burns, talked about mitosis. We will dive into histology, do some review stuff with histology tonight at 5. Any last minute questions before we call our session for now? Good. Kai is asking, will the reticular layer be damaged in a 2nd-degree burn? I believe the correct answer for that one would be yes. 2nd-degree burn starts to damage the reticular layer of the dermis. We definitely damage the papillary layer of the dermis, but we start to damage the reticular layer as well. Ariel asked how many questions. There are 50 specific questions, but like the homework assignment, some of those questions will have more than one part. So you're going to have basically 50 groups of questions. In terms of number of facts that you're providing, it's probably a little bit closer to maybe 100. I think it's worth 90 points. So maybe consider it more like 90 questions. But, you know, on the homework assignments, each of those drop-down menus counts as one question. So keep that in mind. 75 minutes for the exam. Take your time with it. Make sure you have a stable internet connection so that it doesn't die when you're taking the exam. And again, if you have any issues where it kicks you out, contact Proctorio first, because they should be able to get you back into that exam. How are we feeling right now about the midterm exam? I know we still got some reviewing to do, right? What are our thoughts? Gloria's sweating it. Jacqueline's ready to get it over with. Yep, I feel that. Keenan's not impressed, right? Vanessa's a little bit better. Good. Here, let me get a whiteboard here. Let me give you guys my encouragement penguin for any of my friends who are going to take the exam this afternoon. Make you a little encouragement penguin here. Hey, we're looking good today. Look at this. While I'm drawing this penguin, please, if you have any last minute questions, let me know so that I'm not completely just wasting your time. All right, well, my chat has gone silent, so I am going to call it a day. Remember that I will host a histology review specifically tonight at 5 o'clock. So plan to come at 5 o'clock. I will record that, get that posted. I'll get this posted as soon as YouTube's done processing it. So good luck with your studying. Try to review those tissues, try to review some of this stuff before class tonight. But good to see all of you here this morning, and maybe I'll see a bunch of you again at 5. So have a great afternoon. I'll stick around for a little bit longer. And then we'll call it a day.